So what if I wanted to figure out the probability of certain traits being passed to the offspring? For instance, will my child have a dimpled chin? Or can two straight-haired parents have a curly-haired child? Or will my child have a widow's peak like Keanu Reeves? Or will my child have hair on their fingers between the knuckles? Or will my daughter have attached earlobes or free earlobes because free earlobes hang earrings better? Or what if it's as simple as I grow the pea plants that the monks make soup from, and I found that the monks really don't like pea soup when there's yellow peas because the yellow peas make it bitter, and I'd rather have the green peas which make it sweet. How can I control what the offspring will be so that I only get green peas? Now, those are the questions that a man named Gregor Mendel, who was an Augustinian monk, asked while he was tending the garden at the monastery he worked at. Now, Mendel was a teacher. He was a math teacher. He was well versed in statistics, probability. He studied physics. But what he became most interested in was the heredity patterns, how traits were passed in the pea plants. So while he was assigned to teach, he was also assigned to tend the garden, and from that garden grew his interest in how he could control the outcomes of heredity. Now, Mendel did most of this work pre-Civil War, so we're talking about a time frame from 1840 to 1880. So this information was gained and gathered during a time when he had no clue or concept of genes or DNA. He did not have the benefit of Rosalind Franklin's Photo 51 or the model built by Watson and Crick. But still, many of the findings he made about the statistical nature of outcomes we still use today. Now, Mendel was fortunate because he used peas, and peas gave him several great benefits. First of all, their generations are very short. He could see the outcomes over several generations in a short period of time. Not like humans, where it takes up to 15 to 20 years before you can see what the traits of the offspring will be, but he could generate many generations in one growing season and let him gather a great amount of data very rapidly. Second of all, peas have very distinguishable traits. Their traits are very defined. It's either a tall plant or a short plant. There's no middle size. There's no medium. Seeds are either green or they're yellow. There's no in between. Not like with humans where you have, many different forms of eye color. You have green and brown, and you have hazel, and you have gray, and you have blue. With that many traits, it would have been very difficult for Mendel to do the studies he did. Lastly, it's very easy to control the pollination, and Mendel was painstaking in his control and understanding of monitoring the pollination of the flowers. Now, basically what Mendel had to do, painstakingly tedious, he had to take the pollen off of the male part of one flower, transfer it using a paintbrush to the female part of another flower. Before he would do that, he would remove the male parts of that flower, so self-pollination or wind pollination or insect vector pollination would not take place, and he would therefore control his outcomes very easily using this format. Now, what Mendel did is he first isolated plants that he knew the traits for, and beginning with a pure tall plant, and what is meant by pure is he knew that the offspring from that plant would only be tall. He would then pass that or cross that with a short parent, knowing that short parent also was pure, knowing that their offspring should all be short. He called these the P generation, P for parents, and basically the question is, what kind of outcome we will get? And from this, we would expect to get medium offspring, but that definitely was not the case. Now, when Mendel took these two first parents, pure for tall and pure for short, the outcome of those parents he named filial 1, or F1 generation. Filial comes from the Latin word, which is filius for son, or filia for daughter, and that's why we use F1 to denote this generation. Now, the strange thing is all of the offspring of this generation are tall. No medium-sized plants, no short plants, only tall. This is how Mendel discovered which traits were dominant and which traits he called recessive. Dominant traits, of course, mask or cover up the recessive trait. So in this case, we know that tall is the dominant trait to short, and therefore when a tall and a short plant are crossed, all of the offspring in that first F1 generation come out tall. However, when Mendel crosses his F1 generation, the two tall plants, we see that the F2 generation has an outcome of 3 tall plants to 1 short plant. The tall still is the dominant trait, but because the parents both carried the recessive information, there is the possibility of a recessive short plant in the F2 or filial 2 generation. So, 3 tall offspring, 1 short offspring. Why? This is what Mendel came to understand the dominant versus recessive genes and how they worked. So quickly in review, the parent generation, a pure tall plant and a pure short plant, produces the F1 filial 1 generation, all tall plants, but when you cross the F1 generation, the all tall plants that carry the short recessive trait, we end up with 3 tall and 1 short in the F2 generation. So this comes to be known as Mendel's law of dominance, that in an unchecked population, the dominant trait will appear in a ratio of 3 to 1, or 75% of the population demonstrating the dominant trait. Now, the interesting thing about this is what the statistics and probability show mathematically. Mendel always didn't get population rates in these numbers. So there are rumors out there that Mendel may have fudged on some of his statistics, on some of his data. But over time, we now know that this is pretty much true, that in an unchecked population, a population that has not controlled one way or another, we will see the dominant trait at a rate of about 75%, or 3 to 1. Now Mendel took a lot of notes, diagrammed out all of his generations, was very specific in taking diagrams of flower color and pod shape and whether the plants were tall or short, his measurements. He took copious amounts of notes, and we have this history and we can fall back on it to understand Mendel's processes in coming up with this study. Now here are some very important vocabulary that we must come to all understand and agree with. We must understand how to use this vocabulary, because once we look at our own crosses and how we're going to calculate outcomes, we need to make sure we're all speaking the same language and understanding these terms. So first off, the word allele, A-L-L-E-L-E, that's a representation of the trait. So for one of the traits for pea plants, axial flowers, flowers on the sides of the stem, that is a dominant trait. So we use the allele, capital A. For the terminal flower, flowers at the end of the stem, this is the recessive allele, and we use a lowercase value of the same letter. So lowercase a is terminal. So the alleles are the letters we use to represent a trait. The dominant allele is always a capital letter, and the recessive allele is always the lowercase version of that same letter. Now, The genotype, that is the actual pairing of the alleles. We can also use a wording to represent that. The phenotype is the actual outcome, what the plant demonstrates or what we will see. So the genotype A, we call that homozygous dominant. Homo meaning same, zygous from the zygote. That means the sperm and egg joined together to form the offspring. So a A is homozygous dominant. The dominant trait will show through, so these flowers will be axial on the side of the stem. Axial is the phenotype. A, this is called heterozygous. Heterozygous, hetero means different. Again, zygous refers to zygote. Now, when it is heterozygous, A for the genotype, A, the dominant trait will show through, masking or covering up the recessive trait, so these flowers will also be axial. Lowercase a, we call this homozygous recessive. Again, the term homo, H-O-M-O, means same, zygos from the zygote, and these flowers will demonstrate the recessive trait. The recessive trait being terminal flowers, flowers at the end of the stem. So the genotype, this is the allele pair or group of an individual. So this is the genetic information for the organism. The phenotype is the physical outcome of the allele group or pair, and these traits are present in the organism. So, does a witness to a crime give the genotype or the phenotype to the police officer? Of course they'd give the phenotype, because you couldn't see the genes, you couldn't see the alleles inside the individual. Okay, so let's review really quickly. Genotype, A, this is the genotype homozygous dominant. The phenotype would be the recessive trait axial flowers. For the genotype A, this is known as heterozygous, and this phenotype would be axial flowers because the dominant trait will show through in a heterozygote. For the genotype a, this genotype is homozygous recessive, and when it is homozygous recessive, we are looking at a phenotype showing the recessive trait, and in this case that would be terminal flowers. Now, as I said, Mendel was very fortunate in that pea plants had very distinguishable traits. Here are the 7 traits that Mendel worked with. Height of the flower, tall is dominant to short. Seed shape, round is dominant to wrinkled. Seed color, yellow is dominant to green. Seed coat, green is dominant to white. Pod shape, inflated is dominant to constricted. Pod color, green is dominant to yellow. And as we've talked about already, axial flowers are dominant to the terminal recessive flower. Now some other key vocabulary we should be aware of. P1 is the parent generation. Gametes are the sex cells, sperm or pollen for the pea plants, ova or egg. When the sperm and egg join, the pollen and egg join, we get a fertilized zygote. This will create the F1 or filial first generation. And then from the first generation, we get a second generation F2 or filial 2 for the second generation. One other law that is key for our understanding is known as the Law of Segregation. And we must understand that during meiosis, when the cells split into the gametes, whether it be sperm or egg, when the alleles separate, they separate independently. So just like a coin flip, each time you flip a coin, it's heads or tails. Just because you get a heads today doesn't mean you have to get a tails tomorrow. So these gametes are all independent, like flips of a coin. This is Mendel's law of segregation.